Welcome back, and we'll talk about it a little bit. I was sick on Thursday, sorry about that, and we'll dive into that. I had a cold, COVID negative, and I felt really bad that day. The next morning, Friday, I woke up and I was feeling a lot better, and it turned into kind of a chest thing, some congestion. I still have a little bit of that, but I'm pretty sure it was a classic cold, and far beyond it already like that. So we also canceled the lab class. If you're in the lab class, we meet this afternoon. Make sure you take a look at just a couple of Brightspace announcements because we got to jump ahead. Then here's the next thing. The Pre-Med Club is launched, and it's going to have its first meeting this Thursday at 5 o'clock. If you can't make it, make just sure you join, and you'll be on the email list, right? So you'll get a chance to hear what's going on, and if you can make it to the events, you can, and so forth and so on. That'll be great, okay? So then I want to just put a plug in for this. I already did it, but it was kind of out of place. I help organize a research internship at Maine Health Institute for Research in Scarborough each year. And in March of this year, I'll start making announcements, signs and emails to students and stuff like that of the applications for the coming school year. And so that will be apply in March for next school year, September through May. It lasts for the whole year. And this is just an example of what the research groups look like down there. And there are usually a dozen or so USM students down there. Sam is down there. Joe has been down there. And we probably have somebody else in this course that's been down there. And it's the best. It's just the best research internship. It's part of Maine Health. So it sounds like you might be interacting with patients. You're not. It's all pretty much the same. Whatever the topic is, it's all biomedical research, and they're using mice as molecular genetic model systems and studying them in a research lab with test tubes, and the topics are all exactly like what we're talking about. It's down at that molecular genetic level, learning about human health and disease. And it's just a fantastic opportunity, and I have a little bit more of that. So it's paid, and the students get this much a year, $87.40 for being down there during the school year. I think USM is almost unique among colleges and universities in that USM pays students to do internships off campus. It's not any topic. There are some areas that are special focus because it's part of a pot of money called the Maine Economic Improvement Fund. And we have had so much success with students down there who have graduated from USM and gone on to get jobs in Maine or go on to schools outside of Maine, med school, dental school, so forth, grad school, hopefully come back to Maine at some day, so the Maine Economic Improvement Fund is psyched about the work that's getting done there. And I said, let's just boost that internship stipend up. And if you're interested in that and you're going to be around next school year, let's talk about it, okay? So from Thursday, there are these announcements. Normally, I don't talk about the announcements in class because they're the announcements. So I'm just reminding you, you got to pay attention to them. They come out as emails. It's a Brightspace page about announcements, and this one is, I'm sick. I'm sick, but I made a video. And you have to watch that video, and you have to keep up. If you've taken my courses before, you know I record the meetings, and I say then and now, I'm just going to start deleting them. You have a week to learn the material and to take notes that will be your study guide of that material. And if you've taken my courses before, you know that I'm so busy, a lot of times I don't get around to deleting them. And so they're just hanging out there for the whole semester. My plan is always to do that. I have a little more time this semester, maybe. So I plan to start deleting them after a week. They're up there. So you have a week to participate in that class meeting. Either you're here and you sign up like that. So that's the purpose of the attendance sheet. Who signed up? And even if you're here, after you go home, you might be like, I got to go back and watch that one part because I was spacing out, or it was complicated, or I talk fast, right? Because I'm rolling, because we're recording this like that, right? And sometimes I just check to make sure that's working, and it almost always does. Okay? Or you didn't sign up, you weren't here, and I don't care. You don't have to notify me that you're not in class. Your car broke down, you're sick. Don't worry about it. But in the week after our class meeting, you're required to take notes, take pictures of the notes, and send them to me to demonstrate that you're participating in the course. And that's described in the syllabus. It's not part of your grade, it's just part of the requirements for this course. Is try to make the class. If you don't, you got to follow up. And follow up everybody within a week with that class meeting because anticipate it's just going to go away. Okay? So then here's another announcement that's just more recent, and that is, oh, and last week the recording was important for that Thursday. You should take notes because it'll go away, but you're not required to send me notes of that because you didn't miss class, I missed class, right? You only have to send me notes when you miss class. You should take notes on the book too. You don't have to send me those notes, but you know, the reason that you're sending them to me is partly to demonstrate you're participating, you're part of the course, but also to try to get in the habit. We're going to have an assignment today in class to try to get in the habit of drawing this stuff out because it's all physical. It's mostly too small for us to see, but there's lots and lots of ways that the scientists can probe the molecular and atomic details of life. So that's what this is about. That recording was as important as the other recordings, and so get it done because we roll forward. I'm happy to go back anytime and answer questions, but I am hoping that we can do a lot of vertical learning, stack it up. We do it spiral learning. We go over and over stuff because it's hard to absorb. But I'm hoping that you can really learn it because it's the best. So that's our part of our syllabus that explains that requirement. And you can check that out. Remember, you're supposed to watch the syllabus. And so this is what we're going to do this week. Get in the habit of writing down questions and bring them to class, whether you're looking at the book or re-looking at the videos that recorded, or you're just, you know, in another class and you're like, this person is talking about the things differently. And bring them to class. That's really important to clarify. Oh, somebody else is trying to teach you it this way, and you're like, okay, kind of challenge me with that, or any reading or website, like that. So get in the habit. So we'll have this assignment in class today. Try to get in the habit of being active, right? Not just holding your book or not just listening to the lectures in your car, right? So then we'll have that assignment in class. And then if you miss these assignments or quizzes, they're always announced ahead of time. If you miss, you just miss. There's no makeup for this stuff. We'll have a quiz at the end of class today. I could take time to set my timer. We'll stop at 10:30. So you could set a timer too. And if spacing out, just say, ding, 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 or let your clock go off, a phone call, right? We'll have it at the end of class today. It's shorter answer questions based on the prerequisite videos. Then we're going to have another class, a meeting at the end of another quiz at the end of class Thursday. So bring those questions to class. We'll have a time in a minute to just talk all about it because we really want to be solid on that starting material. Then you're reading chapter 1. So write that down, learn from that. I talked about them a little bit in class. I kind of breezed through chapter 1 because chapter 1 is kind of a rangy introduction to cell biology and molecular biology. And but you should try to pick up some of that and not in, I have to learn everything, but try to practice what a biologist practices, which is, I'm looking at this information and it's too much for me to learn, so I'm going to try to identify what's most important and next most important. And the more time I study, I'll get more solid on what I think looks like the most important thing here, and I'll get more understanding of the next most important. Because that's all about why biology, as far as I can tell, is a field of expertise. It's because biologists know a lot of stuff, and they're faced with way more stuff to understand, and they don't try to understand it all. They skip around and try to target and actively learn what is the most important stuff. So then the last thing is this idea that we'll talk a lot about in our course is people learn by recalling. That's how you learn a song, is you recall it, you repeat it, right? And so we got to build that up. We're not going to do that concept chat this week because we're going to have these quizzes. But normally at about an hour into class, we stop and we chat about, well, what have we talked about so far today? Because it's all about recall. And as you walk out of class, it's very good to spend time going, well, what did I just hear about? Or when you read the book, read a page, turn a page, and just look around and say, what did I just read about? Or talk to a dog and say, oh, guess what, Maisie? Oh, Maisie's paying attention. Good job, Maisie. You can participate in this course. If you have a question, get a paw. But that's important. This is a good setting to try to learn about how you learn and what's the best way that you learn. And the questions we've talked about. So, this is a good way to have things look, whether you're in class or at home. So we'll do some of that. This is what we're going to be learning, so you can draw that out. We're going to draw these out today in just a couple minutes. And so that's what we're up to. This is just some images of some students that built some assignments. And so you can probably recognize that as the cell cycle. And in our course, this is one of the levels of the cell cycle that we'll learn, and we'll build that. So, make some sticky notes or something, decorating that with the molecular details of the regulation, and that's the 4th chapter we'll get to. So we're doing intro chapter, we're doing chapter 7 about transcription regulation, which is about the clay model, videos, and gene expression. Then we jump to chapter 16, and that's about cell signaling. And then chapter 17 is a cell cycle, and we're going to crank into it. And you can see, how could you keep this organized? How does somebody keep information organized? And the way they do it is they take notes, but not just linear notes, not just pages and pages of notes. It's good to spread out the learning and decorate it in ways that are distinctive, because when you're recalling those images, hopefully. So that's where we're at. So let's pause here, because at any time you can ask questions, but certainly if you have questions about, what's going to be on the quiz, clarifying that for today or Thursday. And I guess I should have brought a glass of water, because I do have this. I don't feel like I'm infected. I feel great, but I do have some scratchiness in my throat. And I have to leave my phone turned on because the heating people at our house trying to figure out why the heating system is breaking down. So that's a reason for that. Maybe my phone will do stuff. Yes.
Juice boxes if you want one.
I'll have a juice box. Extra credit.
Well,
It's
I
a
have
lot.
to carry them, so if you ever want one.
This is like the 4th juice box I ever drank, because I didn't grow up at the right age, the right time. And they're super cool. But yeah, so comments or questions?
At one point in the video about orthologs, you refer to insulin protein information as collinear with the DNA sequence.
Uh-huh.
And I'm just wondering what you mean by the word collinear.
So it's a cool concept in molecular biology, and that is, look at any protein, and it's folded up. It's a 3-dimensional structure, and in biology, always form dictates function. So you can start up at the macroorganismal level and look at things like ecosystems and niches, and you can look at the organism and you can see, different species of birds have wings that are different shapes. So penguins don't have wings for flying, they have them for paddling, like that. And so form follows function. We have different shaped wings for diving, soaring, and paddling, like that, And that's always true. Drop down to this molecular level. Every protein has a 3-dimensional structure, and that is what confers its function. So insulin, when any protein, like insulin and any other protein, gets synthesized, it fairly spontaneously folds up to a 3-dimensional structure. And that's its function. So when we say, there's an insulin homolog in humans and dogs, it's got the same function. It must have the same structure, like that. Structure determines function, and so that's that. Now, we all know that that 3-dimensional structure of a protein is based on something that's always true about proteins, and that is you could take that and you could stretch that protein out, and it'd be a line of amino acids. So that's a line, so linear information. So that 3-dimensional structure of every protein can be stated by just listing the amino acids, because the amino acids will fairly spontaneously fold up into this 3-dimensional structure. And that's fascinating. I tell students, it's like, imagine that you have a sweater, and the way that sweater got made is you just went to the store and bought a ball of yarn, and you took it out and you just cracked it, and it stretched out, and then it spontaneously folded up into a 3-dimensional sweater. Because the yarn had these little twisty kinks to it, and when allowed, outside of the store in that ball that it's packaged in, they just fold up into a sweater. It's a fascinating fact, and not one to take lightly. That's the linear information. And then, to your question specifically, well, that amino acid sequence, the primary amino acid sequence, the actual just simple chain of those amino acids, that is held in the protein's gene. So every protein has a corresponding gene. We have an insulin protein, we have an insulin gene. And the gene is linear. And collinear just means two things that are both linear and comparatively linear. You can line them up next to each other, and it's two lines of information. And chemically, they're very different, nucleotides versus amino acids. But they confer the same information. So in fact, the DNA that we inherit from our parents is just this line of DNA, but when it comes to protein coding regions, the line is like that sweater. It has all the information to fold up into the protein. The three-dimensional structure is held in the DNA. Yeah, like that. So that's collinear. Yep. And that's always true, right? Another thing to say about it, just to mention this, that's kind of something that a lot of times people just kind of roll over, and that is, for every cell on our planet, it has to make its own proteins. There's very little protein sharing between cells on our planet. When we eat protein, we digest it down to amino acids, and those amino acids go to our cells, and we use those and other amino acids that we can synthesize to make our proteins. So we could eat beef protein or bean protein. We don't use those proteins, even if the beef happens to have some insulin in it. Even that does, and a diabetic needs insulin. They would not use insulin from the food they eat. You can't eat insulin because your body's just going to digest it. So that's a key concept. And each cell has to make thousands of proteins. And every protein has a corresponding gene. Yep. To just put a little underscore on that, something that's true about us is that we only make insulin protein in the pancreas, just in the B cells, some of the cells in the pancreas, do we make insulin protein. To do that, we have to transcribe the insulin gene. We inherited the DNA, there it is. So we have to transcribe that. So in the pancreas cells that make insulin protein, we can detect that protein in an experiment. Yep, it's there, it's being made. And we can also detect the insulin mRNA, because that's there. And both the insulin protein and the mRNA, and all proteins and mRNAs, are kind of unstable. So if a gene is being expressed, like the insulin gene in the pancreas, it always has the protein detectable and the mRNA detectable, and it's constantly producing them. So another thing that's happening is the insulin gene is being transcribed by RNA polymerase into RNA in the insulin secreting cells of the pancreas, and it's just constantly doing that. The RNA polymerase is landing and another one's landing, kind of like a busy airport, and they're just constantly producing that, and the gene is on because it needs to make the product. So it turns out that in humans, every one of our cells has the insulin gene DNA. So that's really sort of a surprise, because we think of evolution as being a very efficient process with no waste. And so when you first hear it, it's a really startling surprise. Every cell has the insulin gene. It will never be used anywhere else. It's only activated in those B cells of the pancreas, ever. Yeah? And almost every one of our cells has every gene. And that's true for every plant and animal on our planet, that they have all the genes in every cell, but they're just not using them all. As a result, a big story of life and the complexity of multicellular life is regulation of gene expression. Oh, all the cells have all the DNA, but we have the pancreas, and we have the brain, and we have the muscles. They have all the genes, all the genes, but they're only using a subset of them like that. It's life is super cool. Molecular biology is the best, right? We should just party. Yep. So that's a question and kind of a long answer, but we're launching into our course, right? So more questions. They could be about the quiz. They could be about Thursday's quiz. Yep.
I was wondering, so paralogs and alleles, is it like allele like at an individual basis while paralogs are at a species base?
Yeah, so we have three terms that we're going to focus on, and a little bit more than that in today's quiz. And the terms are alleles, paralogs, and orthologs. In each case, the definition is we're comparing two genes. And the first one, alleles, is the two genes are the same gene, but just a different version. So an allele is one version of a gene, and there could be another. In our room right now, we all have the insulin gene, but we have some different versions. So we would talk about the alleles of the insulin gene, human insulin gene. Yep. And so that's a story in genetics and evolution, and we really want to jump into the understanding of allele. when we get into the chapters about cancer biology, typically there are new alleles that are problems. The gene got broken, and it's a new allele that is going to cause cancer. Yep. So that's alleles. And really important to appreciate there, we can roam around and talk about any gene and any species, but alleles are just focused on that gene and that species and the variety that is present, So then the next one that jumped to is orthologs. And an ortholog is a pretty dramatic claim, so that some biologists are not very comfortable using that word. And instead, instead they will use a term like, oh, come on. Usually this is sufficient to turn the background to, from this to white. But I have another, oh, I know what's going on here. Yep, so that's going to go like that, I hope. Let's see if that solved it. Well, we can go with a black background, we just have to choose some good colors. That's kind of fun. Okay, so the first term we talked about, alleles. And now we're going to talk about this term ortholog, and then we're eventually going to talk about this term paralog. And for ortholog and paralog, they're going to make bold claims about evolution. And the claims are going to be that they're about something that happened hundreds of millions of years ago, and we're not for sure confident of that. So there's another term that some scientists like to use that's just a more general term, and it's called a homolog. So we'll add that in, but we're going to go right for it because this concept of an ortholog is true, and it's real, and it's really significant to appreciate that and just claim it. That's what's going on. So the definition for an ortholog is really straightforward. Does anybody know what the definition of an ortholog is?
Two different species that share a common ancestral gene.
Yeah, so now, not one alleles are one species. We're talking about humans, we're talking about cats, right? Orthologs, we're going to talk about a gene and compare it using two species. So an ortholog is a gene in two species, any two species. And we looked at that really carefully, and there's something about it that makes us confident that those genes must have a common ancestral gene. So two genes in two species, or a gene in two species that is understood to share a common ancestral gene. So what's an example of that? So human insulin and dog insulin, and it goes like this. People have studied human insulin and dog insulin, biochemists and cellular biologists, they know it's doing the same thing. It's the same thing. It's got the same function in both organisms. Not surprising, we're closely related. We're both mammals. All mammals have insulin. It's doing the same thing in all mammals. In fact, all vertebrates have insulin. It's doing the same thing in all vertebrates. So we know that the structure of that insulin must be really similar because it's got the same function, So we're like, yeah, this looks like the same story. In our course, we'll look at examples of other proteins where we're like, yeah, that's doing exactly the same thing. It's just the same thing in these different species, and we're not surprised because they're closely related. Yeah? Okay? So then, the next thing for to claim it's an ortholog is you say, well, let's look at the DNA sequence, and let's look at it in dogs and humans, and now, currently, there's a tremendous amount of DNA sequence data. And I'll just describe that briefly because it's a big part of our course. So in the 1950s and prior to that, there was no DNA sequence information known. It was only in the 1940s that the first molecular biologists proved that DNA was the heritable molecule. So there was no sequence. Right? So the insulin gene, did they know the sequence of that? Nope. They actually learned the sequence of the insulin protein in the 1950s. Not the DNA, the protein. So this is a story of in labs and biochemists trying to figure out how could we look at the individual letters of DNA, A, G, C, T. But it's pretty clear it's just the pattern, just the order of those letters that differs between species. So you got to be able to read it. So it was in the, really, if you just go ahead and you talk about the date, you know, the decade, like the 1950s, well, none. None of that sequence was done in the 1950s. They discovered the structure of DNA, and they're like, we're psyched to look at the sequence, but they couldn't figure out how to do the biochemistry. So then in the 1960s, scientists actually figured out a little bit ahead of time to figure out a little bit about how to read the letters in RNA. And so there was a little bit of RNA sequencing. That means just reading the letters, because that's all that matters is the A, G, C, T pattern. In the 1960s, the decade of the 1960s on our planet, the amount of DNA sequence that was read by all the scientists in the world was just 100 or so, just a small number. And then the 1970s, scientists, microbiologists really came up with a good way to read the sequence. And so in the decade of the 1970s, on the planet, it's an estimate, but there would be like thousands of nucleotides read total. That's a problem because each human inherits 3 billion letters from each of our parents. So we got to read a lot better than that. But yeah, no, you know, that's the best we could do. And people are excited about it. The people that read the sequences, they got Nobel Prizes. They're just like, good job, you know. The 1980s, companies introduced automated sequencing instruments. So kind of like it's a robotic process and let's get an instrument to do it. So the sequencing jumped up, the technology jumped up. And right at the same time, computers were coming on board so they could start storing this data. And there's really a close match to how computers exploded in our society and how DNA sequence ramped up. Because you got to store that information, right? And you know, it's hard for younger people to understand, right? Like, how did I find rock concerts when I was a kid, right? My big brother and I would just get in the car, and he had actually made a little cone vase of clay in high school art class, and we'd ask people out the window, where's the concert venue in your town? Yeah, so you're like, hey, was that the Stone Age? And I'm like, no, 1970s, when we were having a good time. Yeah, so it's ramping up, right? So 1980s, maybe millions of nucleotides, nothing like all the human sequence. 1990s, ramping up more. The whole planet, a few billion read in the 1990s. And then 20 years ago, those companies introduced a really good improvement in the automated sequencing technology. And so now, starting 20 years ago, we're able to read the whole human genome, but it costs, millions of dollars, right? Very expensive. But this automated sequencing started to increase the capacity and decrease the cost, and that's just exploded like computers explode, right? We can store it, we can sequence it, and so we went from, tens, hundreds, thousands, millions. We don't know anything yet, And it's only 20 years ago we're starting to know something about this stuff, right? And now it's trillions per year. And so everything could get sequenced. All those COVID variants, they're just sequencing those because the virus is just a short piece of DNA. It's just like candy, just sequence them all, So it's really startling how transformative it is. So anyway, we're interested in the insulin genes in humans and dogs. We can easily compare their sequences. In fact, it's a public website where any person can go in and just line them up. And you line them up, and the computer program that lines them up is this BLAST program. I talk about it a little bit. And it's just like a Google search where you give it a starting term. In our case, it's the amino acid sequence of insulin. Insulin is a short protein, but it's still 110 amino acids long, right? And we're just going to use that as our search term. And then we're going to search against all the dog's genes. And just ask the supercomputer, line them up. And it's a supercomputer because it's a big job, but you can just do it on the web. It takes it 10 seconds, and it ranks them just like a Google search. And it's like, this is the best match. And it's dog insulin gene. And there are two numbers that matter. One is the percent identity, and it's 90%. The amino acid sequence in dogs and humans is very, very similar. We're not surprised. Yeah? And then there's another value that's called the expectation value or the E value, and that gives you the likelihood of that match. And that's the number that we key in on when we want to underscore this and say, oh yeah, these are orthologs. These have a common ancestral gene. Because the E value is a probability value, and the way you'd state it is, we're going to do 110 comparisons. That's how long the amino acid sequence is in insulin. So imagine you start out and you're going to represent human insulin, right? And you're like, I'm going to take this coin, I'm going to flip it 110 times. I'm going to write them all down. And then your friend is going to flip a coin to represent the dog match, flip 110 times. And imagine that in that flipping, your friend matches your flips 90%, more than 90 out of 110 matches. And you would be like going to a casino. You were like, we're magic. This is crazy. It's so unlikely, right? Because what's the likelihood? And it's like a half, right? The match is a half, then a half times 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 a half, right? And it's not a perfect match, so that you missed a couple in there, but you can work out that probability, and it's just astronomical, right? It's crazy how well that matches. So here's the key thing. And it's just a feature of life on Earth that just happens to be true, right? And it's that every cell has to make the proteins it needs, and those proteins are linear chains of amino acid, and the DNA confers that, and we can get that DNA sequence information like candy. It's all day long. Just can't go, 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 go. And we get that sequence information, right? And so we're not flipping coins. The match that the BLAST is doing is, it says, this is the first human amino acid. If it matches with the first dog amino acids, that's not a half, because DNA codes for 20 different common amino acids. So matching in one position is 1 out of 20. And then 2 positions is 1 out of 20 times 1 out of 20, 1 out of 400. Less than a 1 in 100 chance just to match twice. And we matched 90 times out of 100. And that's the E value. And the E value is a little bit different than this probability I'm explaining, because the E value considers how many genes the dog has in the process. But it's basically that. And so you'd say, what is the chance that the human insulin and dog insulin sequence could match that well? And so you take 90 and you take 120 times 90, go, do that. And it's not, 1 out of a million or billion or trillion. That would be 1 out of 1 with 6 zeros, 9 zeros, 12 zeros. This one is 1 out of 1 with 55 zeros. It's never happened on our planet that a dog and a human were different and then became that similar. It's just never happened on our planet, in the history of our planet, or anywhere in the universe. There's enough things in the universe, the big universe, to have had that happen by chance. To have them start out differently and converge to be such similar sequences. If they started out differently and they converged to have the same function, they wouldn't have that same sequence like that. Not that match. It's just astronomically impossible. And so that's the thing that provides sort of the stake in the heart of the argument for the ortholog. Because you're like, what do you think? What do you think? And you kind of shiver as you ask this person, what do you think happened? And what the story is, well, we can talk about organisms. Oh, dogs and humans, they both have fur, they both have, a womb to bear the young, they nurse their young, they have a lot of commonality, they're mammals. We have a lot of pieces of data that indicating they must have a shared common ancestor, right? Lots of individual pieces of data, very convincing. When we drop down to the gene, we're not talking about the organism. We're talking about something called the human insulin gene and the dog insulin gene, and we don't worry about what they're in or whether that has fur. We just look at that comparison and we say, those had to have a common ancestral gene. And they become slowly a little bit different. There's no way they were different and became that similar. They must have been the same and slowly got different. And so that's the claim of the ortholog. The ortholog is 2 genes in different species that had a common ancestral gene. Because you're just like, go ahead, think about it. Oh yeah, it's a little propaganda. You guys, where's the, let's get an American flag in here. And sing to it. Yeah, so it's super cool, and that's like a molecular biologist, wheelhouse. That's their deal. Is just like, let's look at that. And so we'll say ortholog, 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 ortholog. Some people like to keep it looser. Homolog just means really looks like the same deal, But we're going to go stronger than that because we do that. And really, every protein in a human and dog genome, they all match up really well. There really aren't hardly really any differences between us and dogs like that and other mammals. Yep. So the next thing to say before you leave that is this. When you see that E value and its tremendous unlikelihood, the reason I say that that is part of the most important feature of life on Earth is because when something is that improbable, it allows you to roam over tremendous evolutionary time. So for example, you say, let's line up the human insulin sequence with a fish. And we've got lots of fish genomes to look. Let's look at them all. But let's look at them one species at a time. Let's look at a trout. So you search for it, BLAST does that, lines them up. And the E value for that, well, first of all, the match drops to like 50%. Only about half the amino acids are identical between a human and a fish. We already knew, well, they're distantly related. They don't have fur, they don't have limbs, They are vertebrates, so they have insulin, it's doing that insulin job, but they're really distantly related. The last common ancestor between us and fish is a long time ago. Yeah, but it is 50% identity, And when you look at the E value, it's still 1 over 1 and 23 zeros, That just never happened, right? So that's what allows. And insulin is a short protein, 110 amino acids. Most proteins, they range a lot in size, but most of them are like 300 amino acids long. Flip the coin 300 times, and match it like that. And so it's just no problem to identify orthologs in all eukaryotes. When we get to that cell cycle chapter, and I showed all the little stickers, those are all orthologs. And there's just no doubt that the same protein is doing the same function regulating the cell cycle in every plant and animal and fungus and single-celled eukaryote on our planet. Because the numbers are big because they're normal-sized proteins. And so it's like a 30% chance of 300 amino acid protein, and that's like 1 out of 1 with 30 zeros. It's just crazy how DNA provides just really, really simple, convincing evidence, with you can't argue about it, about the relationship between the genes and by, extension, all the organisms. Oh yeah, like that. Yeah, so that's another long answer, but it's just so cool to walk forward, when I'm taking a class in molecular biology, and it's really the car mechanics of all life, where you're like, car mechanics are like, we can look at the mower, look at the nuts and bolts on that mower. Everything that, every engine and stuff that's got nuts and bolts, they're really fundamental. So, big part of that. Okay, other questions people have about anything at all? Yes?
I was wondering, do you know the word origin for the prefix for ortholog and paralog?
That's a good question.
Keep
No.
it straight in my head.
Yeah, no, I don't. But yeah, other questions people have. I'd love to say I don't know. Yeah, one time before I lived here, I was in Seattle, and the building was really tall, so we had to ride the elevator. And there was a grad student named Mary, and one time Mary said to me in the elevator, "You have male answer syndrome." And that means like when you ask a guy a question like, "How do you find the mall?" They just will tell you even if they don't know. And so I'm proud that I don't have that. Somebody says, "You know what that is?" And I was like, "No, nope." And people are like, "Whoa, are you scared to do that? Because it's your job to know all this stuff." And I'm like, "Yeah, nope." Also, I'm not scared. So yeah, no. Other questions people have about anything at all? Okay.
I suppose when they're seeing how long something diverged between two species, if something's strongly conserved, how do you take that into effect for if you're looking
Yeah, that's a good question. So there are some things that are really important, like in the process of having the insulin gene. That's really important. If you have a mess up in it, you're probably dead. And evolution is a filter to filter out organisms that have those problematic mutations and kill them. And so the more important functions tend to be more similar over the years, and people will say that's called more conserved. That's an evolutionary term. And that just means they're staying similar more because they're so important. And that's true with those cell cycle regulators. We can find those orthologs because they were important in the very first eukaryotes. They're how you regulate the control of cell division. You don't mess that up, like that. So yeah, so that's a part of it. And you don't calculate that into you don't work that into the calculation. You make the calculation and then you say, wow, that's remarkable. These are clearly orthologs. The people who discovered those proteins, they were studying the yeast that causes bread to rise. And they won a Nobel Prize. Yep. And one of them worked at a cancer research center in Seattle, Fred Hutchinson Cancer Research Center, has a Nobel Prize there for working at a cancer center studying yeast. At one point, that scientist, Lee Hartwell, had gotten the most American Cancer Society funding of anyone on Earth. He's studying cancer using the yeast that causes bread to rise. Because it was so easy to see defects in the cell cycle, the control of the yeast. They would, have errors in their control, and he could study it very easily in the 1990s. Yeah. Yep. So that's something that you get the data, and then you're like, wow, this is really interesting. Yep. Yep. Other questions people have? So let's do this. We're going to jump into this homework, or this in-class assignment, and we won't get it all done today, but it's going to be based on these videos. And so that's fine, because we can work on this some more on Thursday. And again, the goal of it is to get in the habit of sort of drawing this out, writing it out, because the concepts are straightforward. They're just hard to talk about instead of drawing them out, okay? And so what we're going to do is we're going to draw 3 genes. We're going to focus on 3 genes. First, just 2. But what the 3 genes we want to talk about are a series of genes that are needed over time as a precursor to muscle in any animal on our planet that has muscles. When it's first early embryo, it doesn't have muscles. It has precursor cells that are called myoblasts, and this is about when the myoblast changes into a muscle. So these are orthologs of gene products, proteins on all the animals on our planet that have muscles. So lots of people studied fruit flies, and that worked out just fine. And the 3 are the first gene. They're all code for proteins. The 3 proteins are MyoD, and its function is a transcription factor. And one of its genes that it's going to activate is the second, and it's the MyoD gene, myogenic gene. And then myogenic gets activated by MyoD, and then the myogenic protein is a transcription factor, and its target genes are muscle genes. And we can name them, but they're muscle proteins like actin-myosin and a bunch of others. So 3 genes in a row, and the first 2 are transcription factors, and their function is to activate the next gene. So it's like that. And we're going to talk about those. We're going to draw pictures about them, okay? So you're welcome to jump up, because this is back to like kindergarten, where you're like, I need a juice box, dang it. Mrs. Griffey. And you're welcome to jump up, get other markers, get more paper. And there's tape up here too if you want to tape some pieces of paper together. So if you don't have those, or anytime you need to replenish them, just come on up. Okay, so this is what we're going to draw. So the first thing we're going to do is we're going to talk about just 2 of the genes. And we'll draw them left to right because there's a timeline here. So there's the first gene, and there's the second gene. And the first gene is going to be the MyoD gene.
Are we going to fit the 3 genes on one piece of paper?
We're going to use a variety of pieces of paper. Yeah, but the first one, you could turn a piece of paper sideways and put it on there, about like that. So 2 genes, you just draw them out. And you need to learn the terms for each gene. And so one of the terms is the transcription unit. So draw a little box like that. It's just the DNA sequence. It's just the letters, but in fact that works as the transcription unit. And these are genes, so they must have a transcription unit. And you can put a little number there and have a little legend. That's the transcription unit of those 2 genes. And these each have that. So then, the next thing is that they're genes, because there's a transcription unit, and the transcription unit, and you can draw a little color in here, the transcription is going to get transcribed when the gene's expressed. And the big protein that does that is RNA polymerase. So that's the workhorse in this gene expression story. So we got RNA polymerases there. And I mentioned earlier in the class, when a gene is on, it's making several of these. And so that's RNA. Just as a side comment, I've never read this, but I've been a molecular biologist a long time. And here's just something interesting to think about. I believe that the only function of DNA ever is to store information. It's just a storage molecule. And as a result, that means it's a lot like a computer memory. It's just storing that memory. And it's a lot of information. I mentioned humans have 3 billion nucleotides in their human genome. But it's just storing the information. Then we want to use it. And so, on a computer, you have all the files, and they're stored there, and you just open the ones you want to use. And that's like the memory is being output, right? So the next thing is, I haven't read this, but I believe the only way to output information from DNA is to transcribe it into RNA. So RNA is the only output on our planet of DNA information. It first gets transcribed in RNA. Now, the next easy thing to realize is when you want to use a computer, you don't open all the files, you just open the ones you need. And so that's what's going on here, is some of the information is going to get output, and it's a limited information. This is like a file that you open. So you're opening and outputting that information, and you don't open it all up, okay? So what are their files called? The units of information, you open it, and that's the gene. So part of the definition of a gene would be it's made of DNA, it's physical, and the transcription unit is there. That's the part that's going to be output. So that's the first part of a definition of a gene. And it just fits with your understanding of how a computer works. You don't open them all up, and you don't open all the information. You open up portions that you need. And that's what a gene is. Really simple. There's a little bit more to the definition. How do you know when it's going to get opened up? And that is, well, next to the transcription unit is this regulatory region. So every gene has two parts, a transcription unit and a regulatory region. And so we can just circle that, maybe a different color. So somewhere nearby, connected, is this regulatory region. And in our course, that's going to include promoter and one or more enhancers. So you can put those in and label them. That's part of the regulatory region. And there could be some more exceptional details to that, but that's the regulatory region. And so two parts of the gene. The regulatory region is one part, and the transcription unit is the other part, in a really sensible way.
And so genes aren't discrete units in space, they're intermixed with each other.
They can be. So we didn't say that the transcription unit and the regulatory region have to be completely separable. Lots of times they can overlap, and that's fine. And the fact that they can overlap and have some different features is one of the reasons why some people find it difficult to define a gene, but it's this. The next thing to say is that people don't use the same terms for the transcription unit and the regulatory region. That's too bad. If you're taking a class in microbiology, they don't use those terms. They talk about an operon, they talk about an operator, stuff like that. That's too bad. Because this is a universal definition of gene, because it's just really obvious right now, right? Okay, you're going to have the information, and you're not going to use it all at once. You're going to use portions, and of course you have to regulate the output, right? And so for that regulatory region, it's worthwhile, at least on the side, to note this. It only regulates three things with regard to the output, and you just memorize this and say it for the rest of your life. It regulates where, when, and how much.
And that's That's a regulatory region.
Yep, the regulatory region is function is to say where, when, and how much is made. That's what it's doing. Here's an interesting fact of all genes on our planet, is there really only, when you look at an individual gene, there are really only 4 categories of mutations that a gene can have. One would be, the mutation would be in the transcription unit. That's going to affect the output, the product, the output. So we could have a mutation in the insulin gene, and it makes a defective protein, right? That's one category of mutation. And those are the other 3. You could have mutations that regulate where the gene gets expressed, when, or how much. There's more to say about mutations and the details of them, but when you draw a gene, it's good to point that out, that those are really the 4 functional elements of the gene, is the product you're going to make, and when, where, and how much of the product you're going to make. Okay? So that's part of our drawing, like that, okay?
And Dave, when you say where, you mean which cell type? Which type
Yeah,
of cell?
yeah, where, yeah, where, what, in the pancreas, good, you got the gene everywhere, we're only going to make it in the pancreas, yep. Okay. So the next thing before we probably kind of restart another page is that if RNA is the output, right, that's the output of a gene. It's like opening the file and using it, and there are 2 types of genes. One gene is called a coding gene, and coding genes code for proteins. The other is called a non-coding gene, and they don't code for proteins. When we talk about the output, you're used to thinking about, oh, the output of DNA must be the proteins, but it's not. That's the secondary output. The output is RNA. And there's a bunch of different kinds of RNA. The coding genes code for messenger RNA, because the messenger RNA, mRNA, has information from the ribosome to make that secondary output product protein. We have tons of non-coding RNA genes. tRNA, transfer RNA, that's a product, an output. There's tRNA genes, they're the product. They don't code for a protein. They're non-coding genes. The functional product is the RNA. That's it. Ribosomal RNA, small nuclear RNA, small interfering RNAs, microRNAs, lots of different, thousands of different non-coding genes. So we got to just put that in there to appreciate that we got a bunch of genes in us, and some code for proteins, coding genes, and some don't, non-coding genes. Great. Yeah, again, I think the only output of the DNA is RNA. Lots of different kinds of RNA, but it's RNA. The mRNA is a special class of RNA because it goes on to provide the code for all these proteins. Yep. So now probably we want to just somehow start with a new page, and you could overlay it or something to trace it, because we're just going to draw those genes again. And it's the same 2 genes, and maybe I'm picking different colors like that. And here we can just kind of sketch it in. And so it's important to appreciate that I'll just sketch this in, because I'll be like, you guys know, and you can label the terms as you're learning them. But we're looking at we're looking at 2 genes, and I've drawn here the transcription unit, and we're going to focus on the enhancer. And so what happens? Well, this is the MyoD gene again, and the first step is we're going to transcribe that into what you would call MyoD mRNA, because it's a coding gene. It has a coding region that codes for the codons that are going to be in the mRNA. And that's going to bind to a ribosome in the cytoplasm, and the ribosome is going to translate it into protein. So there's a ribosome, and then we're going to have protein product. And I'm just going to make that the same color, and that's the MyoD protein. And this is happening in the cytoplasm, right? That the translation occurs in the cytoplasm. And now this MyoD, that protein, has to get back in the nucleus, because that's where it functions. So it's formed in the cytoplasm, and it has to get into the nucleus. And maybe, you'll have enough colors to make a line like that and call that the nuclear membrane. So this could be the nucleus, and then the protein forms out there, and it's in the cytoplasm. Okay? And its function is to simply bind to myogenin gene enhancer and activate expression of myogenin. And it really likely has other target genes. When we draw these models, a lot of times we'll just draw one molecule of MyoD, but there's always tens of thousands of the molecules of MyoD. And so if there's more than one gene it needs to bind to, and the enhancer of that gene it binds to, and then other genes too. And its function, its entire function, is the same for all transcription factors, and that is they regulate transcription. And so here's the myogenin gene. It's going to get transcribed into myogenin RNA. And that's going to go out, bind to a ribosome, get translated into myogenin protein. And the function of that is it's another transcription factor. So it's going to go back in the nucleus. And we don't need to draw that gene, but let's just say we will. Let's just draw it. So there's some muscle genes. And the myogenin is going to bind to these muscle genes, and we're going to be making muscle proteins. And there are a bunch of those, so it has a bunch of targets, but there's plenty of myogenin to go around. It binds to them all and binds it and gets it done. All right, so now, yes.
So the transcription happens inside the nucleus, and then the RNA is going to go out to the cytosol, where it's going to meet with the ribosome in the cytosol, make the protein that will then come back into the nucleus to attach to the next gene.
Yep. Okay, so now we've got these two genes, and we're going to start talking about the random mutations that could occur that cause no muscles. So that's the phenotype, is we're going to have some mutations that have no muscles. And those mutations can be caused by a bunch of different things. And one of the things is the mutation could be in the myogenin gene, and it could be in the transcription unit and the coding region, and we could make defective myogenin proteins. The defect is in the protein product. It's one of the categories, and they're just all defective. And they could be defective in a bunch of different ways, but we're not going to get any muscles because there's a mutation in the myogenin gene. Okay? So we could just put a little number by that. That's kind of our first category of a mutation, is right in that myogenin transcription unit. And it's a good one to start with because you're like, yeah, obviously, you don't have that functional protein. You're not going to make any muscles. You're not making the muscle genes, no muscles. So the next category of mutation is we could have the same phenotype and have it here in the enhancer where the MyoD binds. That's the DNA. You can have a mutation. You can have a mutation in the regulatory region. We don't activate the gene. The transcription's fine. We could make great myogenin protein. We're just not making it. So that's a different category of mutation. It's not in the transcription unit. It's not in the product. The product's not defective. There's a problem in just getting it made. Okay? And when geneticists do this, they can look at these two mutations, and what would those be? Those would be two alleles that are different, and the geneticist doesn't know how they're different yet. But when they look at them, they can study it, and they can see that these two mutations are close together on the same chromosome. So these two categories of mutations, they're always close together. They're not on a different chromosome. On the other hand, we could instead have a third category of mutation. It could be the MyoD gene. And it could be in the transcription unit, and we're not making good MyoD protein. Darn it. Doesn't even bind to myogenin. Myogenin's totally fine. It's just a different mutation, not a mutation allele in myogenin gene, a mutation, a new mutant allele in the MyoD gene. And that could be in the MyoD transcription unit, or of course it could be here too. Oh, we're not making any MyoD protein. That's defective somehow, right? So these are different categories of mutations. Now, look at this. When the geneticist compares these mutations that are clustered together near each other on the same chromosome, they find that those mutations are different than these. These could be on a different chromosome. They're typically far, far away. Same phenotype, no muscles. Different location of mutations. So a long time ago, geneticists used these terms, and they said, when we see mutations that seem to be close in this gene, they're going to be called cis. And when they seem to be far away, they're going to be called trans. Okay? And so that's an old-time genetics term, but we get terms out of that. Okay? Here's one. Transcription factor. Transcription factors are proteins made by genes that act, they say, in trans with their target gene. They're somewhere else. It's DNA, it's gene, they're working somewhere else. Then on the other hand, mutations here are in cis. So then we have to take our quiz. So last term today, and we'll do more of this on Thursday, more of this assignment on Thursday. These mutations are in cis. And so the best name for the regulatory region enhancer and promoter is what we'll call it, and it's called the cis regulatory region. Right? Because that's distinguishing the myogenin regulatory region from the MyoD transcription gene, right? These are very different mutations. They're in different genes, they're in different locations. But the cis regulatory region has to be in cis. It has to be in cis with the target transcription unit. They have to be together. They have to be tethered physically, okay? So now we're going to put our stuff away. This is an assignment. You're not going to turn it in because I hope you'll just have it become your note. Your having completed the assignment is simply the sign-up sheet. If you're here, you got 100 on this assignment. Yeah? We're going to do another assignment. Just continue with this on Tuesday. Now we have to put our stuff away. Turn your phone off unless they're working on your house heater. And then as soon as you're done with this, you turn it in and head out. There are two sides, so two sides. One piece of paper, two sides. So don't miss that. I did that once in college and I felt bad about it because it was an exam. And there was two sides of the exam. And I was like, oh, really? Yeah, my friends are like, Champlin, you're such a fool. Yeah, those at the front of the cafeteria. So two sides of this. And then there's short answers. Make them direct, matter-of-fact answers. And we'll have another one like this on those other videos on Thursday. You could tell we were talking about the Thursday videos today. So I said we're kind of working on it today. But we'll dive into it more. We'll have time to talk. And this is our big focus today and Thursday. And we'll move forward. And so when you're done, just turn it in, put your stuff up here, and you can head out.